Well, thank you everybody for being here. Today, before I get started, I just want to thank a few people, primarily my advisor, Trisha, my committee members, Ed and Tim, my lab, and then my family, just because without them, I wouldn't be able to be here today to talk to you about humans and marine nutrients. Now, when we think about how humans impact marine nutrient cycles, we tend to think about images like the one you see here of the Mississippi Delta and the Gulf of Mexico, where human activities cause these excess nutrients to get dumped into coastal waters and create these booms of productivity and eutrophication that you see here highlighted by the green. And so how these human activities impact nutrient cycles have greatly generated interest in the different activities that can affect these nutrient cycles. And today I want to focus on how fisheries are impacting marine nutrient cycles. Primarily, I want to pose the question, how have fisheries altered marine nutrient storage? And before I talk about how one impacts the other, I want to take a step back and introduce each component individually, starting with nutrient cycles. And so for the purposes of my talk today, I will be focusing on what I refer to as the big three of marine nutrients, which is carbon, nitrogen, and phosphorus. And so these nutrients exist in the ocean in both inorganic forms like dissolved carbon dioxide or nitrate, or organic forms like dissolved organic waste. And so they get moved into and around the ocean through geophysical processes like coastal inputs and upwelling, or biological processes like fish excretion. And for the purposes of my talk, I want to focus in on the biological end of things. So generally, we've placed a lot of emphasis on how marine life can impact these marine nutrient cycles just because they strongly mediate the transformation of these nutrients between their inorganic and organic forms. And generally, we've placed a lot of emphasis on some of the smaller critters for their roles in these nutrient cycles. For example, phytoplankton for their dominant roles in primary production, or bacteria for their roles in remineralizing and recycling these nutrients. And so historically, we've tended to ignore the roles that vertebrates have in these nutrient cycles. That is until recently, because now we're starting to place a larger emphasis on all the dynamic roles that larger organisms like fish can play in these nutrient cycles. And just to further demonstrate that, I want to show you this diagram, which I know is quite busy, but don't worry because I'm going to walk you through it. Essentially, what you see here in the numbered boxes is all the different ways that these larger organisms can influence these nutrient cycles. And you have examples like what you see here, trophic transfer of nutrients, or deep sea nutrient export due to vertical migrations, or what I want to focus on today, which is how are nutrients stored within the biomass of these larger organisms, and how are fisheries affecting that cumulative nutrient storage. And so what do we need to know about fisheries to properly understand how they are impacting this marine nutrient storage? Well, let's first start by taking a look at how fisheries have changed over time. So here you're seeing a graph demonstrating the trend in global fisheries over time between the period of 1950 to 2018, and where total annual harvests are represented by million metric tons, as you can see here on the y-axis. And for my talk, I want you to focus specifically on the orange, which is just marine fisheries. And so you can see from 1950 to around this point of in the mid to late 90s, fisheries harvest increased steadily over time until they reached that point and have since plateaued and kind of remained stable, averaging around 80 million metric tons harvested per year. And so what this has accumulated to is that over this near 70-year time period, we have extracted 4.3 billion metric tons of biomass from the ocean just through fisheries. And since that's just such an incomprehensible number to think about, I'll let you know that this is in fact equivalent to the weight of 950 million elephants. And so to remove such staggering amounts of biomass, fisheries have to operate in vast expanses of the ocean. And so here you're seeing a map of total harvest just for the year 2015. And the big takeaway, and what I want you to know is that brighter colors indicate larger biomass extraction, whereas cooler colors indicate lower biomass extraction. And so what I want you to take away from this graph is that not only do fisheries have this extensive global footprint, they also vary spatially in the intensity of biomass extraction. And so all in all, fisheries now operate in over 55% of marine waters and encompass an area that is 4 times greater than that of agriculture. And so now that I've talked to you about how intensive and expansive fisheries have become, I can now talk to you about how they're impacting marine nutrient cycles. Now, fisheries, through their mass removal of biomass, are simultaneously removing massive amounts of nutrients that are stored within that biomass. And we've actually begun to study how fisheries impact marine nutrient cycles, but we've only roughly estimated the extent to which they remove these nutrients within the biomass from the oceans. And so there still remains some glaring knowledge gaps that we have to tackle before we can truly understand their impacts. For one, we have stumbled into this era of using generalized body nutrient compositions to estimate nutrient extraction by fisheries. Essentially, we have these percentages, which are the percentage of each of these nutrients, which is the percentage of a fish's body weight that is composed of each of these three nutrients, and we use those to calculate nutrient extraction. Now, the problem with using these generalized percentages is that they're actually an average based on just a few studies that have only looked at about 10 to 20 species. And so what that causes is that we're applying these average percentages that are based on just a few species to the over 1,000 species that we fish each year from the ocean. And just to further demonstrate the variation that these species can have in their bodily nutrient composition, let me just show you an example from my two buddies, the Atlantic salmon and the black sea bream. So you can just see how much they differ in their nutrient compositions. And in fact, they differ by at least 50% in all three of these nutrients. And so let me not just demonstrate to you how these species can differ in these nutrient compositions. Let me also show you what happens if we were to use generalized body percentages versus species-specific body percentages to calculate nutrient extraction. So again, let's use the Atlantic salmon. On the left here, you see the general percentages which I had previously shown you, and its specific percentages right here. And so say we have taken out about 1 million metric tons of Atlantic salmon over time. If I were to calculate nutrient extraction using each of these percentages, then we get something like this. And so you can see that by comparing these two methods, we've actually slightly overestimated phosphorus, but we've also greatly underestimated the amounts of carbon and nitrogen that we've extracted from the ocean through this specific fishery. And so by accounting for this species-specific variation in nutrient compositions, not only can we refine our estimates on nutrient extraction by fisheries and reduce the amounts of over or underestimating of marine nutrient extraction that we're doing, we can also be able to identify which specific fisheries may be having disproportionate impacts on marine nutrient storage. And so we've not only failed to account for this species-specific variation in nutrient compositions, we've also failed to map patterns of nutrient extraction. And so again, I want to show you this map again, just to again demonstrate that not only do we fish in vast expanses of the ocean, we have very strong spatial patterns in where we fish and how much biomass we remove. So we want to consider how in areas of high biomass extraction, like what you see here in the North Sea or here in the coast of China, how high these nutrient extraction patterns can be, and do they mirror biomass extraction closely? And so to tackle these knowledge gaps, my thesis will be asking and answering two questions. Firstly, between 1950 and 2015, how much carbon, nitrogen, and phosphorus was extracted from the oceans via commercial fisheries? And then where in the oceans have the largest extractions of these nutrients occurred? And to answer these questions, my research will essentially follow three steps. Firstly, I will be using the Watson Fisheries dataset, which is a taxonomically and spatially explicit dataset that has harvest data for over 1,000 species per year for the period of 1950 to 2015. And then based on the species listed in that dataset, I will be collecting whole body nutrient composition data for as many of those species as I possibly can, so that then I can calculate the specific nutrient extraction per species for the full temporal and spatial extent of my fisheries dataset. And so just to give you a glimpse of what that may look like, once again, I'm just going to take the Atlantic salmon here. I have shown you before its specific nutrient percentages, and then I'm showing you here its landing just for the year 2015. And so if we take both of those numbers, we can then calculate how many of each of these nutrients were extracted through this species in just 2015. And I know that seems actually pretty straightforward, but when you tell R to do this for over 1,000 species for a 65-year time period, it doesn't become that straightforward. And also to offer you an example of what this may look spatially, I've once again taken the landings data for Atlantic salmon for the time period of 1995 to 1999, taken its carbon percentage, and basically quantified and mapped carbon extraction through Atlantic salmon specifically for that time period. And so say we wanted to compare how the magnitude of this expansion has changed over time. Well, here you have for the period of 2010 to 2014. And so you can see that a fishery that was once very impactful on removing carbon from a system, no longer as impactful. And so these are the sort of things we can ask and answer using the data that I will be collecting and quantifying. And so you may be asking yourself, okay, why go through all the trouble of collecting all this data, quantifying it per species, and then mapping it all up? Well, for once, we can provide the first detailed and spatially explicit estimates of marine nutrient of fishery-based nutrient extraction. Researchers are constantly thinking of how human activities are impacting marine nutrient cycles, but yet fisheries have gone largely unaccounted for in this research. Our research can provide high quality data that will not only help demonstrate the magnitude of the impacts that these fisheries have on marine nutrient storage, but how by ignoring them, we might actually be underestimating the impacts that humans are having on these nutrient cycles. And not only do we need to consider this in future research, we also need to consider it in future management as well. In the realm of fisheries management, by decreasing the amount of nutrients in a system, we might also be decreasing its overall productivity, which that may lead to reduced fish available for fisheries. And then in the realm of climate change mitigation, removing massive amounts of fish, which are known to sequester large amounts of carbon in their bodies and also contribute to deep sea carbon export, we may be underestimating the extent of the impacts of fisheries on climate change mitigation. And by also considering in management, it is also pertinent that we consider the cost, the monetary cost that fisheries may have on these services. So we place a lot of emphasis, a lot of importance on marine organisms for the ecosystem services they provide to us. And also we apply a lot of monetary value to these services as well to facilitate their inclusion in management and policy. For example, we assign a monetary value to fish as a food resource. But then it may become more prevalent now in the future, especially as climate change continues, to include the services they contribute to carbon sequestration, for example, in the realm of carbon markets. And so our research can help demonstrate the importance of considering these species in those sorts of scenarios, as well as potentially offering a way to assess some of the external costs that fisheries may be having. And so just to wrap up what I've talked to you about today, so fisheries have significantly altered the state of the world's oceans. And while their impacts on marine life have been thoroughly studied, we're only really beginning to scratch the surface of how they impact marine nutrient cycles. And so what my research wants to do is to tackle the knowledge gaps that still remain in this subject and describe just the impacts that these fisheries can have on marine nutrient storage. Because by doing so, we can then better assess the cost that fisheries may have and how we can properly manage for them in a rapidly changing ocean. Thank you. Yep.
So I'm wondering what accounts for the species-specific deviations from the, was it just the amount they're storing in fat versus muscle, or
yeah, it can be a variety of things. There's ontogenetic differences, which we aren't considering. There's, trophic level can be an indicator of that. Species higher up on the food chain tend to have more nutrients stored in their body. Phosphorus is very tied to bony structures, so that can differ a lot. And yeah, there's all types of different mechanisms for how this can change, whether that be ontogeny, diets, trophic level, all these sorts of things.
Do you expect to see differences due to different environmental factors for a given species?
So we're not looking at that, but I have read that that can be a cause as well. Yep.
You talk about Atlantic salmon quite a bit. Are you also considering marine aquaculture?
No, we aren't because since those species are actually even being fed, fish feed from fish that have been harvested from the ocean, then we wouldn't want to, kind of double count the amount of nutrients that we've extracted, even from like wild pen marine aquaculture, for example. So yeah, we are not considering that in our research.
You use the word a lot, impact, and you're looking at how much phosphorus nitrogen is being taken out. But the impact of that seems like to be specific to how much nitrogen or phosphorus you have in there. So the Baltic, for example, for the land is nutrient rich compared to the open Pacific. Will you be able to do the fisheries extracted 3.5% of the nitrogen, or just raw numbers of how much is coming out?
So if I'm understanding your question, you're asking how do we account for nutrient extraction in areas where nutrients are already very rich? Is your
Well, you just have huge differences in how much nutrients you have in different parts of the ocean.
Yeah.
And so taking out 1 million tons from per square kilometer out of the Pacific is going to have a much bigger effect than in the Baltic, where you have so many more nutrients.
Yeah, no, and that's exactly one of the things that we're considering. We are not looking into just maybe the inputs of nutrients in from, external inputs into those systems, but it's definitely going to be something we're considering when we're, discussing our results, just seeing where these hotspots of nutrient extraction are based on where they are, because there are, those nutrient poor systems where removing these fish can actually have a more disproportionate impact on the localized nutrient cycles of that system versus, an area where there's just vast amounts of nutrients going into the system, such that maybe these extractions aren't as impactful specifically to the amount of nutrients stored in that system. Yes, Sarah? Mm-hmm. Mm. So for the bycatch data, we do have bycatch data, but we are not considering it because generally bycatch is thrown back into the ocean. So we don't consider those nutrients as removed from the system. That's just the assumption that we're taking. And it's the assumptions that have been taken in other studies that are similar to what we've done. And for the uncertainties, so in our fisheries data set, when you're amassing these massive, this just a global fisheries data set, there's just going to be uncertainties baked into that regardless. And that's kind of like the biggest uncertainties that we deal with. Additionally, there's just going to be species that we will not have any nutrient composition data for. And so we'll have to find ways to fill that, those gaps in the data set. And we've been discussing multiple ways to do that, whether that be taking averages from the, similar species within that family, or using published nutrient ratios to calculate, if we're missing 1 or 2 nutrients, or just different ways of modeling those nutrients to those species that we just don't have any data for. Yep.
So as we overfish the world ocean, 5 of the catch is normally now, which I would think would be really important. Do you have a way to account for that?
Unfortunately, no, we don't, not in this data set at least. Gordon?
How do you go about getting nutrient data for over 1,000 species? Are you going to go out and like catch a bunch of fish?
No.
And like trying to make predictions?
Yeah, no, Trisha bought me a yacht and I'm just going to go on the ocean. No, so I'm using all sorts of published studies, agency reports, and there's data sets for specific nutrients that have been accumulated, that have been compiled already, more so on the nutritional end of things. So fun fact, you can actually use protein to determine the nitrogen percentage of a fish, because in fact, protein is actually calculated from the nitrogen content of a fish. So when you see like the percentage of protein for a fish, it's actually been calculated based on its nitrogen content. So we can get actually pretty exact numbers for nitrogen. So it's those sorts of things that I basically spend all my time in front of a computer just getting data.
We have time for one more question. What are the implications of the feedback cycles of having largely extracted some of the fish populations in certain areas? What does the feedback look like then on how algal growth and phytoplankton growth, and does that exacerbate water quality?
So that's a great question. I honestly haven't really thought about that or looked into that. I know, depending on the system, removing massive amounts of fish can have impacts on just the productivity of the system, especially in nutrient poor systems where fish are these very important vectors of nutrient supply to the system. So you do see that in those types of systems, we see a decline in overall productivity by removing massive amounts of, like areas like coral reefs, for example, where these species are like consuming fish, let's say, out in open waters and then bringing it in and then just resting over the coral reefs and then just dumping those new nutrients into the system, and that affects a lot of productivity. So I haven't really considered your question specifically, but I know that those are some of the things that, removing fish can do to a system's productivity. Thank you.